there's not a single myelin sheath present there. So this is a large area of demyelination, and it's an area calling out for repair. And if we don't repair it, this is what's going to happen. So if the top picture is a smaller view of that, and you're going to see two outcomes. One on the left, where it's not treated, and on the right, where it is treated. So if you watch carefully on the lower left panel, what has happened, you've lost nerve fibers in that area. And this is the critical part of an MS therapy, and what many people are trying to stop happening. Because once you lose nerve fibers, you probably move from the relapsing remitting stage into secondary progressive MS, or this could happen in primary progressive MS. And the chances of any therapy helping you now are slim to nil. So the idea is, how can we stop this happening? So again, watch lower right now. You may see this, because it's a little hard to see. Yeah, no. Okay. I'll go back. So watch the lower right. You see the blue rings have come back. So this is the restoration of the myelin sheaths, or remyelination. And this is there to do two things. It's there to, actually the next slide talks about it. So remyelination, which is our #1 goal, is there for two purposes. The main purpose, from my perspective, is it's going to restore the conduction in nerve fibers. No myelin sheaths, no conduction, loss of function. Restore the myelin sheath, restore function. Very important, the #1 point. And then secondly, maybe equally important, remyelination is the ultimate form of neuroprotection. It protects those nerve fibers against degeneration. That's why it's so crucially important in therapies. How might you achieve this? And I'm going to tell you in detail how you might achieve this, but this is the summary slide. So the first thing is just what I told you previously. The brain can repair itself. That is called endogenous repair. How can we promote that endogenous repair? A key and extremely important new strategy in treating MS. The second, which is what I've been working on for many, many years, is a more kind of, it's a simpler way of doing this. It's simply to replace lost cells by transplantation. Now the cell type doesn't matter here. We're going to use the oligodendrocyte. Now you've seen it written many times. You can definitely spell it. Whether you can pronounce it is another word, the oligodendrocyte, all right? So we're trying to replace the oligodendrocytes, our #1 goal. So the whole notion of stem cell therapies, this is the introduction to this, is something that I'm sure most of you have read something about. Certainly you know, if you're in Wisconsin, you should know about what's happened at UW-Madison in the sense of embryonic stem cells. It's in the State Journal. It's in Time magazine. You don't need to read Nature. You can read it in all the news sources that you will have access to. And this slide, I think, is a nice slide because it illustrates these 2 points that I just told you about in terms of brain repair. First of all, replacing lost cells, exogenous brain therapies, which you see on the left-hand side with the syringes pointing into the brain and spinal cord. And then on the right-hand side, the alternative means of treating is to promote the cell, the brain's own cells, the spinal cord's own cells, to do the self-repair. And we'll talk about those. The source of the cells is one of our major points of contention. It's again a frontier. Where are we going to get the cells for exogenous therapy? I'll talk a little bit more about that. It remains a big scientific challenge, however. Stem cells, what is a stem cell? Just a quick reminder of what stem cells are. A stem cell, by definition, is one that will make more stem cells, and it will also divide to give rise to the cells that make the individual organs within the body. So a stem cell that resides within the brain will make brain cells, within the heart will make cardiac cells, cardiac myocytes, liver, etc., etc. So that's what stem cells are. They have to be able to make more stem cells and then make other cell types or progenitors that will make all of our tissues. Those are adult stem cells, and we'll talk again about what they are shortly. So this is an MS lesion, and we're going to talk first about how you replace cells and then how you promote self-repair. But the area, the blue around the area, this is myelin. The white area in the middle is a large area of myelin loss called a myelin plaque. And this is the idea, if you see again crudely demonstrated in this slide, is you take cells that are in the syringe, you transplant them into the area of damage that I showed you, for instance, in the spinal cord. Very important part is that these cells divide or proliferate. You can only transplant so many cells, it's a finite number, otherwise you're going to cause some damage. You can't inject gallons of cells into your brain or spinal cord. So the cells must divide. They can't divide infinitely to form tumors, but just enough to give rise to the kind of numbers that you would need to repair that area of the brain. They'll mature, so as I said, like the analogy to the baby maturing, not a great analogy, but the cell must get through these various stages to be able to become a myelinating oligodendrocyte. I'll leave that point at that. So what evidence is there for this? I'm going to show you two bits of evidence. The first is we use animal models. So the only way you can do this is to use animals humanely, ethically in research. The idea that we can do this kind of work in tissue culture, for example, is unfortunately not realistic. So the only way to be able to model treatment in MS is to use models of MS. And in this case, we've taken an animal that has no myelin. And what we're going to do is we're going to transplant cells, and that's because of a genetic disease, but it makes a good way to test our myelinating cells. So under anesthesia, we perform surgery, expose the spinal cord, inject cells into the spinal cord, and then we look at this animal 8 weeks later. So you can see that on the right-hand side, the red arrows indicate areas of interest, which, if you go onto the higher power picture, you see them going higher power from the left to the right. So the area where the cells were injected just below, on the left-hand side, looks like a normal area of myelination. I think you got that point already. You know, this is self-explanatory. This is where you don't have to be a rocket scientist to get it. This is an area of complete repair. If you look at the right-hand part over here, this is what it looked like without any cell transplant. So you completely repaired this area, restored all of the myelin sheaths, and this would extend some distance towards head and tail. The scientific way of expressing that is rostral colloid, but we'll just say head and tail. So this is very promising. This is extensive myelination, but not obviously the whole spinal cord or the whole brain. And we've been working on this for a long time. We've got a lot of examples of this. Other labs around the world have done similar.